Schönen Nachmittag, sehr geehrte Damen und Herren, liebe Kolleginnen und Kollegen. Es ist mir eine besondere Freude, Sie am Tag der Universität zum wiederholten Male wahrscheinlich begrüßen zu dürfen. Es ist heute ein besonderer Tag, der Dies Academicus unserer Universität. Wir gedenken der Gründung der Medizinischen Fakultät als Vorläuferinstitution der Medizinischen Universität Wien am 12. März 1365, vor 657 Jahren. Und es war eine bewegte Geschichte, die unsere Fakultät und auch die Universität Wien hier in diesen 657 Jahren erlebt hat. Heute in der Früh hatte ich schon die Ehre, gemeinsam mit meinem Kollegen Heinz Engel, Rektor der Universität Wien, eine Gedenkstunde mit dem Domkapitel von St. Stephan in der Krypta, wo auch der Gründer unserer Fakultät, Rudolf der Stifter, begraben ist, zu absolvieren. Das ist ein sehr ergreifendes und traditionelles Ereignis. Es erinnert auch daran, dass der Stephansdom ziemlich ähnlich alt ist wie unsere Fakultät. Und man sieht also, wie tief unsere Wurzeln reichen. Heute zum Mittag hatten wir dann eine Gedenkstunde am Mahnmal für die vertriebenen Fakultätsmitglieder unserer Universität. Wie Sie wissen, ist der 12. März ein janusköpfiges Datum. Es ist nicht nur das Gründungsdatum unserer Universität, es ist auch jenes Datum, an dem unserer Universität der größte Schaden in ihrer Geschichte zugefügt wurde, als am 12. März 1938 durch den Anschluss Österreichs über 50% der Fakultätsmitglieder der Medizinischen Universität, vor allem jüdische Kollegen, das Land verlassen mussten, vertrieben wurden und unglaubliche Leiden erfahren mussten. Ich habe bei dieser Gelegenheit auch daran erinnert, dass unsere Universität hier diese Erinnerung hochhalten möchte und das eigentlich ein konstitutives Element unserer Universität ist und auch durch diese 3 Denkmäler, wie man sie betrachten kann, das von Dora Barzilai errichtete Mahnmal für die Vertriebenen. Das ist ein sehr schönes Denkmal, wenn Sie einmal in der Nähe des AKH sind. Es ist eine Tora, aus der eine Seite oder mehrere Seiten herausgerissen wurden als Erinnerung an den Verlust an herausragenden Persönlichkeiten und auch an Wissen unserer Universität. Und im Hof danach gleich das berühmte Monument von Sigmund Freud, das auch noch zu seinen Lebzeiten vom jüdischen Bildhauer Oskar Nemon geschaffen wurde. Und wir haben es genau am Tag 80 Jahre, nachdem Sigmund Freud Österreich verlassen musste, von der Familie Freud zurückbekommen. Und ich war vor Weihnachten in New York und habe mich mit Professor Kandel, den Sie kennen, getroffen, und der war so nett und hat zugestimmt, dass das Center for Precision Medicine, das wir in den nächsten Jahren errichten werden, hier an der Universität am AKH Campus, seinen Namen tragen wird. Also es wird Eric Kandel Institute Center for Precision Medicine heißen, und auch das ist eine Erinnerung an die Geschichte unserer Universität. Wir kommen jetzt zu einem weiteren Highlight der Universitätsvorlesung, die heute ein sehr berühmter, wenn ich so sagen darf, Auslandsösterreicher halten wird, Professor Florian Krammer von der Icahn School of Medicine, Mount Sinai Hospital in New York. Ich werde dann noch ein paar Worte quasi im Vorspann sagen. Er wird diese Vorlesung auf Englisch halten. So haben wir das vereinbart. Ich möchte aber vorab noch ein paar Kolleginnen und Kollegen erwähnen, weil es, glaube ich, in diesem Rahmen wichtig ist, hier auf ein paar Highlights unserer Universität hinzuweisen. Ich möchte Sie einladen, sich auf unserer Homepage die Spots anzusehen. Wir haben 3, oder nicht wir, sondern aus unserer Universität wurden vor Weihnachten noch 3 Einreichungen zu Clusters of Excellence, also Exzellente im Rahmen der FWF-Exzellenzinitiative erstellt, von den Professoren mit großen Teams dahinter, Professor Hackani, Professor Bock und Professor Valenta, zu den Themen, ich kürze das jetzt ab, Neurowissenschaften, Allergieforschung und Precision Medicine. Ich habe mir über Weihnachten diese Anträge angesehen und auch durchgelesen. Ich darf Sie herzlich einladen, sich das auch anzusehen. Es sind ganz tolle auch Updates. Es ist sehr lehrreich und man sieht, wo die Front des Wissens in diesen Bereichen derzeit ist. Und ich bin den Teams und ihren Leitern also sehr dankbar für diese unglaubliche Arbeit, die sie hineingesteckt haben. Und ich bin sehr hoffnungsfroh, dass wir zumindest einen dieser Clusters of Excellence auch tatsächlich an unsere Universität im Laufe des Jahres kriegen können. Dann möchte ich noch einmal erwähnen den Herrn Kollegen Professor Peter Klimek vom Complexity Science Hub, ist vielen von Ihnen aus Film und Fernsehen wahrscheinlich bekannt. Er ist einer der Mathematiker im Prognosekonsortium, das seit 2 Jahren Tag und Nacht die Covid-Pandemie begleitet. Er ist auch aufgrund seiner Kommunikationsleistung heuer zum Wissenschaftler des Jahres gewählt worden. Ich habe darauf schon in meinem Neujahrsbrief Bezug genommen und möchte das noch einmal hier tun. Wir hatten auch 2 Inventors of the Year geehrt. Das waren die Frau Kollegin Gabriele Häusler und Thomas Wrba für ihre sehr innovative Erfindung im Zusammenhang mit Wachstum.at. Bitte schauen Sie sich das auch einmal an, es ist ganz toll, www.wachstum.at. Und dann möchte ich auf die Veronika Fialka-Moser Preise hinweisen. Da haben wir eine Liste an verschiedenen Preisträgern, die ich jetzt auch namentlich erwähnen möchte. Wir haben 2 Kategorien. Erste Kategorie Diplomarbeiten 2022, Dr. Lovro Markovic, Dr. Simon Reichert, Türkan Akaya Kalaci und Dr. Yen Tan. Und in der Kategorie Engagement 2022 Charlotte Rössel, Igor Grabovac, Veronika Riedl-Schlaus, Stefan Riedl und Andreas Böck. Ich habe jetzt aus Zeitgründen die Titel weggelassen. Also herzlichen Dank für Ihr großartiges Engagement und danke auch an alle Jurymitglieder. Und last but not least möchte ich noch auf die sehr erfolgreiche Einwerbung eines WWTF-Grants hinweisen, einer Vienna Research Group for Young Investigators aus dem WWTF durch Sarah Melzer im Bereich Kognitionswissenschaften. Damit komme ich jetzt zur Vorstellung von Professor Florian Krammer und ich werde mir erlauben, jetzt auf Englisch zu switchen. Und ich möchte vielleicht mit einem Brief beginnen, der mir untergekommen ist, und zwar ein Schreiben. Sie können es jetzt nicht sehen und ich werde es aus technischen Gründen nicht in die Kamera halten, aber das Schreiben ist unterzeichnet von jemandem, den Sie alle gut kennengelernt haben, Dr. Anthony Fauci, MD. Tony Fauci is one of the icons of medicine, as you know, and he wrote a letter to Professor Krammer, Florian Krammer, Icahn School of Medicine at Mount Sinai, which reads as follows. So I won't read the whole letter, just a few parts. So Tony here says, on behalf of the NIAID, I would like to thank you for the critical contributions you and your team have made during the ongoing COVID-19 pandemic. Your research has been incredibly important to the fast-paced pandemic response and has supported public health efforts around the world. You are a leading example for both the infectious disease and broader biomedical research communities, and I'm grateful for your unwavering commitment to global public health. So I think there's not much more to add. I will quickly say a few words about Florian's, Professor Krammer's outstanding career. He was born 40 years ago in Pack, so those of you who kind of commute between Carinthia and Vienna know the Pack. So he was born in Styria. He graduated from the, as we call it, BOKU, so the University of Natural Resources and Life Sciences here in Vienna in 2010, and earned an Erwin Schrödinger Fellowship of the FWF, which is kind of one of the first step of, I would say, high potentials to go abroad. And Professor Krammer obviously was aiming for the best of the best, so he joined the team of Professor Peter Palese at the Department of Microbiology at Icahn School of Medicine, Mount Sinai in New York, and has stayed there ever since. Some of you might know the name Peter Palese. Also, Professor Palese is an outstanding researcher. He did his PhD with Professor Hans Tuppi. So Florian Krammer is here in a very high-profile chain of Austrian international researchers. So he stands in a very long tradition. Also, with regards to Mount Sinai, I would like to mention Hans Popper. You know Hans Popper, or the name of Hans Popper. He is also of Viennese origin, and he was one of the, at least intellectual, founders of Mount Sinai School of Medicine. I checked out Mount Sinai School of Medicine, and what I found was an interesting quote, which I think might also be related to Professor Krammer's career. It says, any school may prepare you to make a living. Icahn prepares you to make a difference. So I think this is a very interesting quote, and it, I think, fits very well to Professor Krammer's outstanding achievements. At present, he holds the position as a professor of vaccinology at the Department of Microbiology at Icahn School of Medicine at Mount Sinai since 2019. And so his main research interest, as you know, is related to virology in a very broad sense, but also in immune defenses against different RNA viruses. And he also leads an important and huge collaborative influenza vaccine innovation center, the CIVIC, and has made a large number of other achievements, which I will not mention. I think you got the point. And Professor Krammer, it's a huge honor for us that you give us this lecture. As you probably know, last year it was Christoph Huber, also an outstanding colleague from Mainz, who told us the mRNA COVID vaccine story, and I'm really looking forward to your talk. So good morning to New York City. The floor is yours.
Thank you very much for this great introduction. I'll try to share my slides here. Just 1 second. All right, here we go. So what I'll try to do today is to look at the bigger picture of, SARS coronavirus 2 and beyond, and how we can get prepared better for future pandemics. Before I start, I wanted to make my disclosures. Our institution has filed patent applications for SARS coronavirus 2 serological assays and certain SARS coronavirus 2 vaccines. I'm not going to talk to about these today, but these filings name me as inventor. So the questions that I wanted to address today is, first of all, where are all these pandemic viruses coming from? Second, what can we expect from SARS coronavirus 2 in the future? And then third, how can we get better prepared against future pandemics? And the first point I wanted to make here is that these outbreaks actually constantly happen, right? We constantly have viruses that jump from animals into humans. Here's just a limited list of outbreaks that actually made it to the news. Very often the public doesn't really realize that these things are ongoing. And there's actually many more. So I highlighted here the 2009 H1N1 pandemic and the SARS coronavirus 2 pandemic that started in 2019, 2020. But these outbreaks are very common and they happen all the time. Actually, if you're interested in that topic, there's a mail server that's called ProMed, and you can sign up there, and you will get emails from all across the globe from MDs, from researchers who report zoonotic infections, who report small outbreaks, and you will be surprised of how often that happens. Most of these outbreaks actually don't have consequences because they can be stopped, or they're too small, they don't take off, or they happen in areas where there's actually not enough connectivity so that they can spread. And another reminder that we have now seen this Sars Coronavirus 2 pandemic, but there were other pandemics in the last 100 years that had a lot of consequences. I'm just showing influenza here, where we had a pandemic in 1918, which caused, depending on the estimates, 20 to 100 million deaths. We had another influenza pandemic, H2N2, in 1957, which caused about 1 to 3 million deaths. Another one in 1968, H3N2, which caused another 1 to 3 million deaths. And then the 2009 swine flu pandemic, which caused about 250,000 deaths. And in addition to that, there was, of course, also the AIDS pandemic that is still ongoing. So where do these viruses come from? And the answer is pretty clear. They come from animals. Often they come from rodents. Bats are very often a source. Birds, specifically for influenza. Domestic animals, there's a lot of diseases that domestic animals carry. And we see here that interactions with livestock happen. This picture actually has a reason. There are H3 and 2 variant viruses that circulate in the US in pigs, and during agricultural fairs, where there's close interactions between humans and pigs, they often jump into humans, and they can potentially also cause outbreaks and pandemics. And of course, there's a lot of viruses that are transmitted by ticks and by mosquitoes. Here on the right side, I'm just showing a list of zoonotic infections that happened with influenza viruses in the last approximately 20-25 years. And you see, this is a pretty long list of circumstances or cases where influenza viruses jumped from animals into humans and partially really caused severe disease. None of these really took off, but this happens all the time. And the last examples are really H5 and 8 in Russia, where we had this virus jump from chickens into humans. We currently have H5 and 6 cases in humans in China, with actually a relatively high case fatality rate. And then there's the ever-looming threat of H2N2, which has caused a pandemic in the past and is still around in the avian reservoir. But you know, a lot of these outbreaks seem far away. But I think we have to keep in mind that they are happening also in Europe. They are also happening in Austria. I don't know if you're aware, but we have West Nile virus in Austria. We have Lesotho virus in Austria. The southern part of Austria sees partially a lot of cases with hantavirus infections in humans. And very recently, in the middle of Europe, there was also a filovirus that was identified in pets, and that's the Lovio virus, which is a close relative of the Ebola virus. And so this was identified in Hungary in 2016 in Schreiber's pets, which actually have a fly range that also basically includes the southern part of Austria. So these viruses are not necessarily really far away. Another point, and that's an important point, is that these outbreaks, the number of these outbreaks is increasing for many reasons. One reason is that there's, of course, better surveillance and diagnostic methods. So we really have the technology to detect them. And so that's one reason why we find more of these viruses and more of these outbreaks. But also the world is better connected. And so, you know, virus infections can spread quickly. I'm showing you here on the lower right side, basically airline connections. Globally, this is from 2009, so there's probably more now. But a virus infection that is acquired somewhere in Indonesia can make it in less than 24 hours to the US, and you can have transmission chains all across the globe. The other point is that there are more people And that means that there are more chances of getting infected. And if there's more people, we also have more domestic animals because we have to feed these people. And of course, that also increases the human-animal interface, and that increases the chance that a virus jumps into humans. And then, of course, there's climate change. We now see that certain mosquitoes that we didn't have in Austria, for example, in the past are now showing up. Certain tick species are showing up, and they can be competent vectors for viruses that haven't been in the moderate climate zones before. And of course, climate change also leads to behavioral changes, and that can lead to more exposure to certain viruses as well. And another point is natural habitat destruction, right? If you have a virus that circulates in non-human primates somewhere in the Amazon, but nobody lives there, and there is no humans that interact with this wildlife, there are no infections, right? But if you start to cut down trees and if you start to cultivate that area, there might be interaction, and that might facilitate the virus to jump. The other problem that we are seeing, and this is really a policy problem, is that our response to outbreaks is often reactive. So basically an outbreak happens, the news starts to report about it, politicians pick it up, and then there's money poured into the response to the outbreak. Often then the outbreak is contained with non-pharmaceutical interventions, and then the money for working on vaccines, doing the research is actually drying up. And we have seen that multiple times with pandemic influenza, with Zika, with Ebola, and with Sars in 2003. And it would be much better to actually have a proactive approach where you do the work beforehand, and when an outbreak happens, you're actually already prepared. And I'll go back to that in the end of my presentation. I just wanted to show one example of this, and that had a lot of consequences. So there were a lot of Ebola vaccine candidates that worked really well in non-human primates. But there was no commercial interest to further develop these vaccines because, Ebola outbreaks happen in rural areas, in small villages typically. They can be contained, and there's certainly no market for an Ebola vaccine, right? But in 2014, we saw an outbreak of Ebola in West Africa that actually grew bigger and bigger and bigger. And in the end, more than 20,000 people were infected and more than 11,000 people died. But the vaccines were not ready. They could have been ready, but when the outbreak was growing, then only first steps were taken to actually test these vaccines in clinical trials, to look at their safety, to produce them in the right quality. And these vaccines came too late to actually make an impact in this outbreak. One of them went through a phase 3 trial in the end that had results. And now we have 2 Ebola vaccines that are licensed, and they can be used in outbreaks, but we certainly were not prepared for that outbreak. And this is just another example of a reactive response instead of being prepared. But now let me focus on coronaviruses, because I think everybody's interested in that right now. So just a few basics. Coronaviruses are called corona because when you look at them under the electron microscope, they appear like a crown. And so you have these spikes that are protruding here. This is actually the spike protein of the coronavirus. The genome of coronaviruses is relatively large. It's the largest genome that we find in RNA viruses. It's often more than 30,000 base pairs. We have a lipid envelope. This is shown here. That's where the glycoprotein sits in. And the virus particle is approximately 120 nanometers in diameter. These coronaviruses are nothing new. We know about them. They circulate in animals. We know about turkey coronavirus for a long time, but they also circulate in humans, and they cause up to 30% of all common colds. And we have 4 of these seasonal human coronaviruses. These infections are usually relatively mild, and I'll get back to that towards the end as well. But then we had in the past, and you could see them as warning signs, 2 zoonotic coronaviruses that caused major outbreaks, and both of them are relatively dangerous. The first one is Sas Coronavirus 1 from 2003, and the second one is MERS coronavirus. I just wanted to, before I tell you a little bit more about these 2, I wanted to show you a phylogenetic tree of the coronaviruses. You hear a lot talk about universal coronavirus vaccines right now, and so on and so forth, but the diversity of these viruses is humongous. What I'm showing you here is phylogenetic tree that actually emphasizes the beta coronavirus genus, where we have, as part of that, our subbacoviruses, and that's where Sas Coronavirus 2 sits. Here is the Omicron variant. Here is Sas Coronavirus 1. But this is part of a much bigger genus of beta coronaviruses that also include the MERS coronavirus and then the 2 human seasonal coronaviruses. And in addition to that, we also have the alpha coronaviruses, the delta coronaviruses, and the gamma coronaviruses. So there's huge diversity here. And when we talk about variants, this distance here is minimal compared to the phylogenetic diversity that we see within all the coronaviruses. Now, a little bit about these 2 zoonotic coronaviruses that have caused issues in the past. The first one is severe acute respiratory syndrome coronavirus, SARS-CoV, or how we call it now, SARS-CoV-1. And this caused an outbreak that started in 2002. The epicurve is shown here on the bottom. What happened here is there was an outbreak that started in the Guangdong province in China. And China didn't report the cases. It wasn't clear what was causing it, but then cases occurred in Hong Kong in January and February, and Hong Kong reported this to the WHO, and an investigation was started. You can see the epicurves here, and the virus was then exported to a lot of different countries, including to countries in Europe, but also to Canada and the US. And in Canada, we actually had a pretty deadly outbreak of nosocomial infections with SARS-CoV-2. Now, the interesting part here is that for a long time, people didn't actually know what was causing the disease here. Only in April, so at the last peak of the outbreak, was the virus identified and the genome sequenced, and it became clear that this is a coronavirus. We were very close to an actual pandemic at this point in time. I don't think people realized that. There were about 8,000 cases, again, with many cases in Canada, and about 800 of these infected people died. So a case fatality rate of approximately 10%. It then turned out that this virus came from bats, and the virus jumped from bats into civet cats, which are used as food in China, and from civet cats into humans. This was 2002 to 2004. So the outbreak was basically contained with non-pharmaceutical interventions in mid of 2003. Then there were a few single infections in 2004. The second virus that caused coronavirus that caused issues in the past is the Middle Eastern Respiratory Syndrome coronavirus, or MERS-CoV. That virus is also pretty deadly and has an approximate case fatality rate of 35%. And so this started in 2012, 2013. The first case was reported by an Egyptian doctor who worked in Saudi Arabia. He had an elderly patient who was infected, who had respiratory syndrome. He sent samples to Erasmus Medical Center in the Netherlands, and they found the virus and determined it's a new coronavirus. And then you can see we had these waves in 2014, 2015. Most of them happened in the Middle East, and it was also determined where this virus is coming from, and it's actually coming from camels. So in camels, MERS coronavirus causes relatively mild infections, basically sniffles, but camels are kept as animals for camel races and things like that in the Middle East, and people closely interact with them, as you can see here on the right side. That's when the virus jumps into humans and can cause disease. The red peak here is interesting because that was an outbreak in South Korea. And this was caused by a traveler who went from the Middle East to South Korea, was infected, went to the first hospital in South Korea, wasn't diagnosed there, went to the next hospital, and in the end visited 4 hospitals and infected a lot of people there. And so there was an exponential increase in cases in South Korea, and they were able to contain this with non-pharmaceutical interventions. But again, we came pretty close to an outbreak that could have taken off and could have caused a pandemic. Again, with the case fatality rate of 35%, relatively serious incident. But now let's focus on SARS coronavirus 2. So December 31st of 2019, China reported to the WHO that they had several cases of pneumonia with unknown etiology in Wuhan, in the Hubei province. So this is here on the Chinese map. And there was an association to the Huanan seafood wholesale market. And that market doesn't just sell seafood, they sell all kinds of meat there, including live animals. And then retrospectively, there were reports that actually the outbreak started earlier. You can trace back cases to the beginning of 2019, and some of them were actually not necessarily directly connected to that seafood market, but there's now much more data. And I think with the last 2 or 3 scientific publications that came out, we can really tie the outbreak to that seafood market and to transmission from animals into humans. And this was the start of the SARS-CoV-2 pandemic, basically. It didn't take long to identify the virus in this case. So the report to the WHO was made in the end of December 2019. And on January 10th, Professor Yongchen Zhang from Fudan University identified the virus, sequenced the genome, and shared the genome with Eddie Holmes, a virologist in Australia, and asked him to just post the genome online, right? So they didn't go through all the red tape, they just released it. And actually, Yongchen Zhang got reprimanded for doing that. But of course, this helped a lot. This helped research, this helped vaccine development. So for me, he's really one of the heroes of this pandemic. When you have the genome, you can actually do phylogenetic analysis, and you can find out what the virus is related to, what type of virus it is. And so it became pretty clear, and we did that too. We downloaded the sequence on January 10th and built a phylogenetic tree. It became very clear that this new virus was closely related to SARS coronavirus from 2003, very closely related. And so that's why it was in the end called severe acute respiratory syndrome coronavirus 2. So that's the virus. And the disease itself was called coronavirus disease 2019. And that naming was done by the WHO. So one is the virus, the other one is the disease. How does it look right now? So these are data from yesterday. We're at 450 million official infections. We're at about 6 million official deaths. You see the case numbers here on the left side. This is global case numbers. And the deaths on the right side, again, this is global. You see that there were several waves globally. And the last wave here stands out. This is Omicron. Omicron also caused a lot of deaths, but if you look at the size of the wave here and the size of the wave here, you can see that there's a different proportion, and that tells us already that Omicron is not causing as many deaths as proportionally as other variants and waves before. But we'll go back to that. That doesn't mean that Omicron is harmless, not at all. And as you can see, there's a lot of deaths here, unfortunately. These are the official numbers, but there's also estimates of the death toll that are based on excess mortality. And so The Economist did one modeling of that in December, and they estimate that between 11 and 21 million individuals died of COVID-19. Their average is 18.1 million. There was recently, I think yesterday, a paper published in The Lancet that actually estimates 18.2 million. So it seems that the actual death toll is much higher than the official death toll. So how can we prevent getting sick, getting infected? Of course, immunity helps us to defend our bodies against virus infections. And first of all, we knew a lot about immunity to coronaviruses from previous studies, from previous viruses, but also there was a lot of knowledge that was gained in the first few months of the pandemic. And now it's very clear that we get good antibody responses against the virus if these antibody responses are directed against the spike protein. They can actually neutralize the virus. So the spike protein is essential for the virus to bind to our cells, specifically this receptor binding domain shown here in red. And so if antibodies bind here, the virus can't bind to our cells anymore. And so that means it's neutralized. And we also know that there are robust T-cell responses that after virus infection. They are mostly CD4 T-cell responses. There's a relatively weak CD8 T-cell response, but this can protect us as well. And this immunity forms the basis for the vaccines that have been developed. I won't go through the details here, but as you know, there's now a number of vaccines that are available. They had very high efficacy against the original Sars Coronavirus 2. And there's a whole diversity of vaccines. We have the mRNA vaccines here, vector vaccines, and now even a recombinant protein vaccine that was licensed in the European Union. And so there's basically 5 vaccines that are available to people in Europe. There's 3 that are available in the US. The 2 are mRNA vaccines and the J&J vaccine. But globally, there's many more. And actually, the vaccines that are used most globally are inactivated vaccines from Sinovac, Sinopharm, and Bharat. But we have now really tools to prevent infections, prevent severe infections, and these vaccines have saved millions of lives worldwide. At the point where vaccines were introduced, and that was first done late in 2020, we also saw variants of concern coming up. And those were, of course, problematic, as I'll describe in the next few slides. The first 3 that we found were the alpha variant, the beta variant, and the gamma variant. The alpha variant was detected in the UK. It had a change in the receptor binding domain and directly at the interface between the receptor binding domain and the ACE2 receptor. And so what this mutation did was it made this interaction stronger, and that made the virus more infectious. The virus this variant doesn't necessarily escape neutralizing antibody responses, so the vaccine still protected very well against it. The beta variant, which was detected in South Africa, by the way, South Africa has an outstanding genomic surveillance system for Sars Coronavirus 2 that's much better than what most countries in Europe or in North America have. They detected this beta variant, and in Brazil detected the gamma variant. They had additional mutations, and those led to escape from neutralizing antibodies. Not complete, relatively minor, but still the virus was able to deal better with neutralizing antibodies and was able to escape to some degree. And then there were a lot of variants of interest that were detected, partially also with escape phenotypes, but none of them really took off in the end. What took off was Delta, and Delta posed really the first problem for vaccines, not necessarily because it circumvented immunity by introducing mutations that so that the spike wouldn't be recognized anymore, more by being more infectious and producing much higher viral loads. And so what I'm showing you here is the epi curve from the US, but it's very similar in Europe. In spring of 2021, the case numbers went down, the vaccination rates went up, and a lot of us were assuming that the situation would now get under control and everything would be better. And I assumed that too. It was a reasonable assumption. But at the same time, we had a wave in India, a relatively severe wave, and that wave produced the Delta variant, and then the Delta variant spread globally, and Delta is much more infectious than the original variants. And so it was starting to spread in the US here in June, and you can see in the beginning of June, it was responsible for about 10% of the cases. And then within a few months, basically, it took over and basically was responsible for almost 100% of the cases. So that was the first variant that was a major troublemaker. And then in November 2022, Omicron was detected, and this went really fast. So on November 22nd, there was a partial sequence from Hong Kong that was uploaded into a database, into GSAID, and this was from a traveler from South Africa. And an evolutionary virologist from the UK looked at that sequence, said this could be a problem, and posted it online on Twitter, talked about it, and on November 23rd, so the next day, there were already sequences from Botswana and South Africa that became available and basically showed the same mutations. And so this was designated as the B11529 lineage, and that really stood out because of the large number of mutations, including in the spike protein. On the 25th, there was a press conference in South Africa. They had a wave, and they determined that most of that wave was caused by this new virus. And by then, we already had case reports in Israel and Belgium. Basically, that already told us that the virus was spreading globally. On November 26th, the WHO announced that this is now a variant of concern, and the rest is basically history. We had the first Omicron, or the first Omicron cases here in New York in this beginning of December. By mid-December, this was already a relatively severe wave. Retrospectively, the virus has also found, or the sequences of the virus have also found already in October in Nigeria. Just to show you what the difference is here between these variants, we have on the right side spike with the mutations that Delta has indicated. We only have 2 mutations in the receptor binding domain. There's a few others outside of the receptor binding domain. And on the left side, we have Omicron. And as you can see, this is really a monster. We have 15 mutations in the RBD, the receptor binding domain, many more mutations outside. And if you're familiar with where neutralizing antibodies bind and the epitopes of these antibodies, just by looking at the sequence, you could already tell that all of these epitopes had changed, and that this virus would most likely escape the majority of the neutralizing response induced by vaccines or prior infection. And so the consequences of Omicron were that double vaccination provides little to no protection from infection. And I'm specifically saying infection here. Infection, disease, and severe disease are different, very different categories. What I say here really means infection. We know that a booster dose provides protection from infection, but this wanes relatively quickly. We also know that reinfections with Omicron are relatively common. This was not the case for other variants. Infection protected well against reinfection. But we also see now that baseline immunity, either through infection or vaccination, seems to protect well from severe disease. And I think this is a very important point that's often understated. The other important point is that Omicron infection in naive individuals leads to Omicron-specific immunity. Our colleagues from Innsbruck have shown that very nicely. There's now other data that corroborates that. But if you had no immunity and you get an Omicron infection, your immune response is Omicron-specific. It might not protect you from Delta. But in general, and I think that's a good thing, is that Omicron causes less often severe disease as compared to Delta. It's comparable to Alpha, actually. But that doesn't mean it doesn't kill people. And unfortunately, as you have seen with the epicurves, it has caused a huge amount of deaths. And just to drive this point home, and that vaccination is still very, very important, I'm showing you here epicurves of Hong Kong and New Zealand. Hong Kong has about double the population of New Zealand, but this doesn't matter here because it's per million people. And what you can see here is on the left cases in Hong Kong and New Zealand. Both have a huge Omicron wave at the moment. So you can see how quickly the case numbers went up and at the same time. And on the right side, you see deaths. And this goes only up in Hong Kong, but not in New Zealand. And the reason for that is that Hong Kong didn't manage to vaccinate the elderly. Only 30% of the elderly in Hong Kong are vaccinated. In New Zealand, it's almost 100%. And this is really the difference here. Omicron can cause very severe disease in people who have no immunity. As soon as you have immunity, your risk is much reduced. The other point that I wanted to make here is a point about when it makes sense to lift restrictions. So what you see here on the left side is the Omicron wave in New York City. So it started in December. We went up to more than 40,000 cases per day. Just to point out, New York City has about the same population as Austria. And then this declined. And here in March, actually on March 7th, we lifted our restrictions. On the right, you have the situation in Austria, right? You see the case numbers going up, and then they go down a little bit, and now they're coming up again. And the restrictions here were lifted on March 5th. And so I think when you lift restrictions, you really have to look at the epidemiological situation and make rational choices. And maybe it's not a good idea to do that when you're in the middle of a severe wave. Coming back to these variants, coronaviruses are an exception among RNA viruses. Typically, RNA viruses don't have proofreading activity. They replicate their genome, they make mistakes, and that leads to mutations. But coronaviruses actually have proofreading activity, so they don't mutate as fast. However, if you have a lot of cases, that means you have a lot of replication, and that gives a lot of chances to the virus to make mistakes and to mutate. And that's what we have in the pandemic. But there's another mechanism at play. What has been seen is that there's faster evolution in immunocompromised hosts who can get infected persistently. Here's just an example from a New England Journal of Medicine paper where a person was infected for 150 days and repeatedly sampled, and you see some of the mutations that we find in variants coming up here. For example, E484K, N501Y, and so on and so forth. And the leading hypothesis right now is that these variants actually come from immunocompromised individuals who carry the virus for a long time. The main question now is, how will the future look like? How is this going to look moving forward? A lot of viruses evolve through this ladder-like pattern, right? So you have one variant, and then that mutates a little and becomes the next variant, and that mutates. And so each of the new variants is a descendant of the old variants. And this is easy to predict, relatively easy to predict, and if you need a vaccine, it's easy to update the vaccine. Some viruses like influenza B, for example, split into two lineages, but then they also evolve in this ladder-like pattern. And again, it's relatively easy to make vaccines against these viruses, even if you need now a bivalent vaccine. But other viruses have a more erratic evolution where these new variants are not necessarily descendants of the old variants. And unfortunately, so far, that has been the case for SARS-CoV-2, right? So all these variants actually come out of the center. They come all out of the wild type virus, right? And that's the problem. So this makes it really hard to predict. If new variants now would all be descendants of Omicron, it would be relatively easy for vaccine updates, but this is not clear yet. And this is also it's also important here to make the point that Omicron just came out of the center, and by chance it was a little milder. The next virus could, land here, and maybe it's less mild and more pathogenic again. So this is really a challenge, and hopefully the virus will evolve in the future more ladder-like. What will happen in the future? We have our 4 human coronaviruses that cause up to 30% of all common colds, and they have really this seasonality, right? You start to see cases in October, you have a lot of cases in winter, and then it goes down. And I think it's reasonable to assume that this will also happen with Sars coronavirus too. That it will assume this seasonality, at least in the northern and southern hemisphere. Just to point out, this OC43, which is one of the human coronaviruses, is a suspect to have caused the 1889-1890 Russian flu pandemic. This is based on phylogenetic analysis, probably jumped into humans from cattle. But over time, it also became, in a way, a seasonal coronavirus, and probably became milder, not because the virus became milder, but because you get infected as a kid and you develop immunity, and when you get older and you would be at more high risk, you already have immunity against the virus. So the biggest question now is what is SARS coronavirus 2 going to do? Where is it going to fall? And I think it can basically fall between the influenza scenario and the human coronavirus scenario. For influenza, we also see seasonal circulation. There's substantial severe disease, and there's an annual vaccine, and we need that vaccine because of the severe disease. For human coronaviruses, we have also seasonal circulation, but mild disease, and we don't need a vaccine. And so the question now is if SARS coronavirus 2 is going to fall closer to the human coronaviruses, which would be great, or if it's going to fall closer to influenza, where we actually need vaccine updates and where we need to continue to vaccinate. But this is something that we can't answer at this moment in time. And then just to also give an update of a positive update about what novel therapeutics are coming and which new vaccines we can expect. There are now therapeutics and prophylactics that are either already available against Omicron, like sotrovimab, which still works, but likely it's less effective against the BA.2 variant, subvariant of Omicron. We already have Paxlovid, which is really a great tool to reduce hospitalization and deaths. And molnupiravir is also in the process of getting approved in Europe. And then there's a prophylactic monoclonal antibody cocktail coming that's called Evusheld, which has a really long half-life. And I think this is going to be a super good tool to protect people who have immune deficiencies and can't mount an immune response themselves. So this is given twice a year and should give long-lasting protection. In terms of vaccines, I think we will see variant-adapted vaccines. I hope there's one coming for Omicron, but this depends a lot on regulatory agencies and pathways and also on viral evolution. There are also a few mucosal vaccines in development, and these might actually block infection and might be more effective in really curb a spread of the virus. And we know from studies this is also important to point out, we know from studies in Israel that a fourth dose of the current vaccines does not really provide much benefit to healthy individuals. And so to close this now, I just had a few thoughts about how to prepare for future pandemics. We have now the situation where a virus emerges and vaccines become available much later, and we have to go through many months of global virus spread, death and suffering, economic losses, partially even societal disintegration. And it would be much better if we would have a vaccine within 3 months that could be used and we could avoid many deaths. And so there is a way to do that, actually, if we really get prepared. We would need to have more surveillance to understand better what's out there. And based on that surveillance, we could select a number, maybe 50 to 100 virus strains from different virus families, and could start to prepare vaccines against these viruses. You could make GMP material, you could go into phase 1 and phase 2 trials, and in the meantime, you could try to understand better correlates of protection for these viruses. And then when a new virus emerges, you can quickly look what you have already as vaccine, and you can look what this virus is related to. And if you made a good selection, you probably have a related virus that's already been tested in phase 1 and phase 2 as vaccine. And you could probably just switch the strain. And then you could produce this new vaccine and start a phase 3 trial right away, because you establish correlates of protection. This vaccine may be licensed within a very short period of time based on the correlate. And so I wrote a commentary about that, but this is also aligned with what CEPI is proposing. There's a 100-day challenge, and I think within 3 months, a new vaccine could be available. Of course, this costs money. Phase 1-2 trial costs about $20 million per vaccine per virus, but you have to put that into perspective. And I just put the number here for the US defense budget, which is $686 billion for 2019. Not saying we should reduce defense spending right now in this situation. This would be very foolish to say, but I think we should make preparation for pandemics part of our defense spending and defense thinking. And just to put this more into perspective, this $20 million for a phase 1-2 trial, this is a Eurofighter, and it costs about $140 million per piece. So that's 7 vaccines that you can bring into phase 2. Of course, I look at this from a vaccine point of view, but we also need other preparations for future pandemics. We need broad antivirals that cover whole virus families. We need a global framework and a concerted plan of action in case a virus emerges. In the beginning of 2020, it was all chaos. Nothing was concerted and coordinated. What also came through was that we need local production capacity for masks and other protective gear. A lot of that is imported, and when a pandemic breaks out, these import routes might be closed, or countries might not be happy to export these things anymore. And I think one of the most important points is also to develop efficient communication strategies to make sure that the population is on board and engages in countermeasures. We see now this anti-vax movement. We see people on the street demonstrating against vaccines, against masks, against countermeasures. And I think we need to find a way out of that, and we need to make sure that the whole population is on board when we have to respond to a new pandemic. And with that, I'll close, and I'm happy to answer questions.
Okay. I hope I'm back again now. Well, so thank you very much for this comprehensive, beautiful, and concise overview of the world of SARS coronaviruses. I have a huge number of different questions here in front of me, and we will now spend the next 30 minutes or so, if you want, in a kind of question-answer scenario. And I would like to start off with your take on a zero-COVID strategy. So in light of also the data you showed in China, Hong Kong, and so forth, is this really a prudent long-term strategy to go for zero COVID like China, for instance, as opposed to other parts of the world?
I think it's worth trying, right? In New Zealand, it worked out. They minimized the number of cases during the initial phase of the pandemic. Once vaccines became available, they made sure that everybody got vaccinated. And now they can't stop Omicron. They have to open up, and it comes in and it does little damage. In Hong Kong, on the other side, we see the same, but the preparation was not that great. Now, of course, Hong Kong is easy to protect from incoming viruses, and so is New Zealand, right? But there are other larger countries that also were relatively successful in keeping the virus at bay, including South Korea, for example. And so I don't know if zero COVID is working, but to minimize infections is, in my opinion, a good strategy if vaccines are on the horizon, and if you think that you can vaccinate a large proportion of your population. If there's no vaccines coming, of course, it doesn't make much sense, and it's probably not helpful to just try to have no cases, because at some point they will come in, right?
Sure. Next question is, so when SARS-2 emerged, it was said that coronavirus don't mutate that much. So obviously this is not the case. So people were a little complacent and saying, well, it's different like flu, and if we find a kind of a vaccine, it will be a very stable situation. And now we find us in a different world. So with lots of different variants, with the need to adopt vaccines and so forth. So is this kind of unexpected, or something you would have seen before?
So honestly, for me, this was unexpected too. Again, coronaviruses have proofreading activity, so they make fewer mistakes, they mutate less than comparable RNA viruses. So it was a surprise, but on the other side, when alpha, beta, and gamma emerged, I think that was the point when people really started to realize, no, this might happen, this will happen, right? So I think, of course, we got surprised, and it was in a way unexpected, but yeah, I think now we have to expect that a variant can come out of nowhere.
So you think more surprises to come, so it's not the end of the road yet?
I think there will be more surprises. The question is, will these be things that we as virologists, we as medical community have to deal with? Maybe we are going to be in a situation where we worry about it, but it's not something the public needs to be worried about anymore. And that's hopefully the situation we end up in. of course, every year I'm worried about mismatches with the flu vaccine and nucleates of H3 that could come up, but that's not something that's on the cover of, I don't know, Corona-Schutzimpfung, right? So I hope it's becoming less and less relevant for the public.
Okay, so what is your opinion on this position that if a virus kind of evolves, so if it jumps from animals to humans and then overcomes the barriers, the two barriers, it typically over time gets less dangerous and more infectious. Is this kind of something we can count on or not?
The easy answer is no.
Okay.
So, of course, if you have a virus that has a case fatality rate of 90%, that virus is going to kill its substrate at a very high rate, and that might be not beneficial for the virus. And so in that case, there might be pressure to become less pathogenic. But with SARS coronavirus 2, there are a few factors. First of all, you can actually transmit the virus to somebody else before you become sick, right? So there's not much evolutionary pressure. Once you transmit it, the virus couldn't care less if you die or not, right? Because you already did the job. That's the first part. The second part is really the evolution that we see right now. We don't see this ladder-like evolution where, you know, you might have a variant like Omicron that's a little bit less pathogenic, and then the next variant that comes out of Omicron might be even less pathogenic. That's not the case. Right now, all these variants came out from wild type, from the center. And so the next variant, if it comes out if it's a descendant of wild type and not of Omicron, might well be as pathogenic as Delta again, or maybe even a little bit more. So I think in terms of what we see now, that there's less and less consequences, I think that has more to do with our immunity, the baseline immunity that a lot of people now have, than with the virus becoming milder. So I wouldn't count on that.
Okay. So many more questions. Decoupling. So could we conclude that the decoupling phenomenon that you also showed with regards to the cases, to kind of ICU and death rates, is not a biological phenomenon of Omicron, but rather an immunological phenomenon of a more immunologically protected society?
Exactly. I think the main factor here is really immunity. And I think the New Zealand versus Hong Kong example drives that home, right? Of course, the more baseline immunity we have, the less consequences a virus infection will have in the population. And ideally, in the end, you have mild infections and very few deaths, right? But I think we have to keep in mind that there are other consequences too that these infections can have, right? It's not just how many people do we have in the hospital, how many are in the ICU, how many are dying. It's also what are the sequelae, right? How many long COVID cases do we get? And that's something that's usually underemphasized. And long COVID, I mean, it's a complex of things. We don't understand it well, but it is problematic.
Thank you. So another question on this important topic. So what should be kind of the leading parameter in pandemic decision making? So I think we have been focused on case numbers for 2 years now. Now there is a little bit of a shift to hospitalization, ICU pressure, and so forth. I also had phone calls with colleagues from ICU departments in New York around Christmas time. So I think they were very worried, but I think you did a great job. If you look at that curve with the steep increase and the steep slope down, what do you think? Is it a good idea to look or to focus more on hospitalization numbers and leave cases away because it doesn't matter so much?
I don't know. I mean, one of the issues that we had here in New York was that at times a third of our hospital staff was sick. during the Omicron wave. And that has a lot of indirect effects too. So I think you need to keep both things in mind, right? And I have to emphasize, we had a lot of deaths during Omicron here in the city too. There were actually fewer ICU admissions, but there were a good number of deaths too. And that's actually, the Omicron wave in New York City was the second deadliest wave that we had in the city. The Delta wave wasn't as deadly, for example. So yeah, I think you need to look at all numbers, but case numbers are also important. And the other problem that we often have is the case numbers are an early indicator, right? The case numbers go up first, people start dying later. And so once you see that you have a lot of ICU cases and you're at capacity, and that's too late to break, right? This is minutes before you hit, or seconds before you hit the wall, right? And I think ICU admissions is not really a good indicator if you want to plan ahead and you want to really step on the brake and make sure that you don't hit capacity.
But looking at the current Austrian situation that you pointed out, so we have this, I think the case fatality rate initially was around 0.5 to 1% roughly. Now we are way below 0.1. People are comparing it with flu and so forth. And so if you look at the curves here in Austria, you see that actually the ICU cases are stable, if even decreasing, whereas we have these enormous levels of daily infections. So is there anything we can make of it?
There's a few points, right? In the UK, because the vaccination rates there are really high, they're now down to a case fatality rate that's comparable to seasonal influenza. That's not great. Seasonal influenza is a really big problem, but it's much better than the situation half a year ago, of course. The other question is, what if you would have a seasonal influenza wave in Austria where we had that many cases, right? What would be the countermeasures there? And I don't think, one of the important points is also we're not necessarily talking about lockdowns here as countermeasures, right? Just before the pandemic started, I think in the late fall of 2019, there were schools closed in Austria. I think it was in Tyrol because of flu outbreaks there, right? So it's not just that you have to have a virus that's more deadly than influenza. And it's nice to see that there is no increase in ICU cases and that, this is stable, but that's not the whole story, right? Again, if we compare it to flu, we also have to compare it to what we would do if we would have that many flu cases, right?
Thank you. So there are many more questions. So when do you expect the approval of mucosal vaccines? Is there anything we can expect from that?
There are a number of developments currently. There's a vaccine that is developed in Hong Kong and in China that is based on influenza, that is a nasal spray. There is an attenuated Sars Coronavirus 2 that has been developed that's now also, I believe, in phase 3 trials. The vectored vaccines, the AstraZeneca vaccine is getting tested intranasally. We have an intranasal vaccine that in Mexico is in phase 2 and in the US in phase 1. So I think if one of the ones that are further ahead gets approved this year, it could become available. But there's a lot of question marks there because it has become harder and harder to do phase 3 studies. You have to do immunobridging. And then the other issue right now is that these vaccines are still all based on the wild type virus. And it might be hard to see efficacy in terms of protection from infection against Omicron. So there's a lot of issues there. And then there is a funding issue, right? A lot of funding agencies are now basically saying, we have vaccines. Why do we need new vaccines? Of course, there has now been a push towards mucosal vaccines again because of the breakthrough infections. So it's unclear. If you're lucky, we might have one by the end of the year, but it might also take substantially longer.
Thank you. So you pointed out seasonality. I think this is also mysterious in a way, but because in some places of the world there seems to be seasonality, and there are other places where there does not seem to be seasonality, like you mentioned South Africa or Singapore is an example, Brazil. What's up there?
Okay, so we have to look at other viruses there too, right? So typically common cold, coronaviruses, influenza, they have seasons on the northern hemisphere and on the southern hemisphere, right? So they occur in winter. That is never the case in Singapore or Nicaragua or southern parts of China. There you have very irregular waves of infections, right? And we haven't really figured out what drives this seasonality yet. This might have to do with temperature, it might have to do with humidity, it might have to do with human behavior, being indoors versus being outdoors. It might even have roots in the microbiome and how the respiratory microbiome might change in colder weather and warmer weather. But I think it's reasonable to assume that SARS coronavirus 2 will follow the course of all these other viruses that in the northern hemisphere and southern hemisphere show seasonality. If that will ever apply to a place like Singapore is questionable. And again, Singapore might have 3 flu waves during a year and they might be distributed randomly, right? So again, if you talk about seasonal coronaviruses or seasonal influenza, we only talk about places that have seasons. And if you're in the tropics, that doesn't really apply. Again, we don't have a good understanding of why we have seasonality, but I assume SARS coronavirus 2 will do the same.
Thank you. That's some good news for Austria at least. So what is your opinion? Yeah, it's an almost political question here. You know this testing story. Austria, I think, tests like 10 times more than other European countries. I tried to take a test in New York. I think it costs around $180 or so. Here it's for free, so we do a lot more testing, and there's this discussion, so how useful this really is. Do you have an opinion on that?
I do have an opinion. I think it's important to know if you're infected or not, because then you can take actions to make sure that you don't infect other people. And without testing, you will not find out how this, if you're infected or not, right? But you're right, Austria tests much more. We have pretty nice testing here in New York too. And actually, if you live in the city, it is actually for free. It's just a problem if you're from the outside. But yeah, I think testing has an important role, but I cannot tell you what's too much testing and what's sufficient testing.
A very difficult question came up here. So do you have a guess for a causative virus for a future pandemic?
There are actually a number of viruses out there that I'm personally scared of. And actually in the past, SARS coronavirus 1 was one of them. And I'm giving a lecture every year at BOKU where I talk about emerging viruses. And I always brought SARS coronavirus 1 as an example where, it almost happened, right? But there are other viruses out there that are problematic. One of them is Nipah virus. That's a virus that jumps from fruit bats into humans. Historically in Malaysia, now more in Bangladesh and in India, in areas there which are highly populated. And that virus has a case fatality rate of above 50%. It goes into the central nervous system, and it has been shown to be capable of human-to-human transmission with a reproductive number below 1, but still. And so if a virus like that would adapt further and cause outbreaks, that would be a disaster. We also have seen, I think in 2019, the first super spreader events with a hantavirus in Argentina. That is also very scary, but there are lots of viruses out there that might be related to what we see now that are unknown yet, right? MERS is another virus that is problematic. And of course, being an influenza virologist, I can just tell you that all the influenza viruses are very scary too. So there are a lot of viruses out there. It's just the question, is there a chance that they transmit? And if there's reproductive infection and further on transmission, there could be mutations and they could become more efficient in transmitting. But again, also to put this into perspective, there have been serological studies in China, in rural China, to look at exposure to SARS-like viruses in the population before SARS coronavirus 2 happens. And a huge number of people there actually have antibody responses, meaning that they were exposed and nothing happened. So they might not have had a productive infection, or the virus might have never spread. And so these things happen all the time. Again, it's hard to predict, but there are candidates out there that we should keep an eye on.
Well, flu isn't so much of a problem as opposed to unknown dangers. At least that's my estimate, because for flu you have a clear-cut procedure. So you have like a number of drugs which kind of help you over the first phases, like neuraminidase inhibitors and so forth, and it is almost predictable that you will have a vaccine within like 6 months or so. Whereas I think for COVID, it was just luck that we got it so far. Would you agree on that? That we could better cope with an influenza pandemic as opposed to all the other viruses you mentioned now?
To a certain degree, yes, because I think we could actually get to a pandemic flu vaccine if needed within 3 months. There's a huge safety record on these types of vaccines with other strains. And as you said, there are drugs, and now not just neuraminidase inhibitors or ion channel inhibitors, but also polymerase inhibitors. So there's a number of things that can be done. The question is, how much can be produced? What proportion of the global population can you protect? And, you know, if you talk about antiviral drugs, it's also a question of how much can be supplied. So I think a new flu pandemic could lead to a very high death toll, but you're right, we're better prepared for flu than for other things.
Talking about drugs, so what is your view on potential drug resistance? So you mentioned those 4 classical anti-Omicron drugs now. Well, in particular, I think that the polymerase inhibitor, molnupiravir, and the Paxlovid. Do you see any kind of problem here that if we prescribe or use it too frequently, we will run in a resistance problem and we might lose it?
We already did in a way with the monoclonal antibody therapeutics, right? Not because we used them and we drove resistance, but because resistance just developed naturally, right? Except for sotrovimab, all the monoclonal therapeutics are basically off the market because they don't work anymore against Omicron, right? There is always the chance that you drive resistance, and for influenza, we have seen widespread resistance to neuraminidase inhibitors, to ion channel inhibitors. So these things can happen. So it's very important that you have several tools, right? And that's the nice thing now with SARS coronavirus 2. We have monoclonal antibodies, both therapeutic and prophylactic, and we can make new ones if there's resistance. And we have polymerase inhibitors, and we have protease inhibitors. And so I think, you know, having this larger number of therapeutics, prophylactics that target different mechanisms of the virus is important. And so I'm not too worried about it. You know, if we lose one of them, I think we can come up with another one, and there are safety nets. So I'm not too worried about it.
So we totally switch off gears. Obviously, there's a polio outbreak in Ukraine. At least this is one question. Is there any concern with that? Have you heard of it?
I haven't heard of it.
It was. It is said it was reported in Time magazine. Polio outbreak in Ukraine. It might be concerning.
I haven't heard about the outbreak in Ukraine. I know that there was a polio case detected in Israel, and that's very concerning. We were close to getting rid of polio, and this comes now back in a way, it seems, right? But we have that problem with many different diseases, right? Measles is the prime example. Measles outbreaks are becoming more frequent because people don't get vaccinated against measles anymore. And I think if this continues, people will find out what terrible disease measles can actually be. But yeah, polio is, of course, very concerning.
One question here asks, so what is the use of further vaccines. So I think that there are like hundreds of vaccine development programs at different stages of kind of development. The mRNA vaccines and all the others we have now seem to be relatively effective. As we know, the protection fades away, unfortunately. This is something we don't like that much. But do you see any kind of niche for particular new kind of vaccines which provide an add-on to what we're seeing right now? Or is it better to say, okay, now I think we have all the weapons in place and we just should learn how to better use them, because otherwise it will get very confusing if we have like dozens of different vaccines?
I think there's 3 points here. The first point is it would be good to have strong mucosal immunity, and we don't have vaccines that do that for Sars Coronavirus 2. The second point is it is not clear how often you would need to get revaccinated, right? If this needs to be done more often, I'm still not thinking about annual vaccination, but more like either when a new variant comes or maybe every few years, like the tick-borne encephalitis vaccine. The mRNA vaccines have a certain reactogenicity. People after the second shot often felt sick and they had to stay home for a day. And so if you find and have vaccines that don't do that, but are as effective, I think in the long run, they will take over the market. It's just a basic fact. If there's a vaccine that works well and doesn't give me side effects, and a vaccine that works well and gives me side effects, I'm going to go for the one that doesn't, right? And the third point is really capacity building. What we have seen, and unfortunately this was predictable, there were vaccines produced and the rich countries were basically buying them and keeping them. And it took a very long time until there were enough vaccines provided to low and middle income countries. What we now see increasingly is that low and middle income countries also have vaccine candidates, and they move them forward. And they might not make a huge impact this time, but they will have the expertise to do it next time, and they might be much faster. And I think this is an important point. I think we need geographically distributed vaccine production capacity. Countries need to be able to make these vaccines locally so that they don't depend on Moderna's willingness to give them vaccines or Pfizer's willingness to export vaccines, right? South Africa, for example, is taking a big initiative right now with mRNA vaccines, but you see that in many other countries too. And I think that's a very important point to make countries independent and self-sufficient in pandemic preparedness and vaccine production.
One question is here is on antibody testing and kind of approaches to measure cellular immunity. Well, do you see any advantage, except for scientific reasons, to do that?
Well, I mean, the antibody testing right now is very complicated, right? Because we have a correlate of protection that you can also, if you model it for a variant, you can also apply it to variants, but it's complicated. On the other side, serological testing is in a way very helpful for people who are immunocompromised, right? We have people here, for example, in our multiple myeloma cohorts, who don't seroconvert, right? You vaccinate them 3 times and they still don't have antibody. And they need to know that, because that means they are still very vulnerable. And scientifically, we also look at their T cells, and in that cohort, they don't make a T cell response either. So I think this can be helpful for patient management in some cases, but because of the complexity of variants, the correlate of protection is not very helpful for managing patients right now. Of course, more is better, and there is a clear correlate with neutralizing antibody and binding antibody. This has been published now a number of times. But again, the slope in terms of how much antibody you need to be protected changes according to the variant, and that makes it hard to use it in practice.
Another question on long-term COVID effects. So long COVID, fear of MS, EBV, even maybe cancer and so forth. So how worried should we be? So it's hard to really grab it because it's kind of an unknown. But how likely would you think are long-term COVID kind of problems? We have followed the field in the field of borreliosis, for instance, where there has been a huge debate, also public debate, and in some cases also an obscure debate. So many different symptoms were bunched together, and it was claimed that people have kind of long borreliosis or long-term Lyme disease. Is this something we should kind of prepare for, or would you say, well, from what you know, it is not very likely that we see a huge problem here?
Well, I mean, one thing is long COVID, right? Which is, again, a whole mix of different symptoms and probably different mechanisms. And this is real, right? There are people who don't get their full lung functions back. There are people who don't get their sense of taste and smell back, which sounds funny, but I think if you lose that, you lose a pretty big part of enjoyment in your life. And so long term, I think that's a big issue. There's what people describe as brain fog, which actually also has been described for the 1918 pandemic, influenza pandemic. So there are real things out there, and there are real issues that patients experience. The other thing is, can we speculate about more long-term effects? Cancer, for example, this is not clear. I mean, there's no data right now. Looking at other human coronaviruses, I think there's no data either, but it doesn't seem that there's an obvious connection. And I hope that this will be the same for SARS-CoV-2. But as you see now, there's nice papers coming out in terms of what different herpes virus infections can do. You never know, right? And it might take many years to really understand what the impact was.
Okay, so I think we've got 5 more minutes and 2 more questions. So I think we're perfectly on time. So were there any attempts to develop a SARS-CoV vaccine before the event of the pandemic we're facing now?
Yes, there were 2 vaccines that were brought into phase 1 after the 2003 Sas Coronavirus 1 outbreak. Both funding dried up in phase 1. There was no interest in doing this anymore because the virus wasn't circulating in humans anymore, which is a typical example of short-sightedness. But one of these vaccines was actually produced by the same company that is now making CoronaVac, and it used that as blueprint. So it's basically very similar. The other one was a DNA vaccine that the Vaccine Research Center here at NIH was developing. For MERS, there were also plans to move ahead and develop vaccines. But I think the most important point was that so when people started to design preclinically vaccines against coronaviruses, they found that the spike protein is often unstable. And so researchers at the Vaccine Research Center, Jason McClellan, for example, they found a way to keep the protein in the pre-fusion conformation, the conformation that you need to get a good antibody response. And so this was done for SARS coronavirus 1 and MERS coronavirus. And that really informed also vaccine design for SARS coronavirus 2. So there was work that was done, but not extensive work.
So last question, anything we can do to prevent transmission from animals to humans in general? So there was this discussions about Chinese food markets and so forth. Anything you can think of?
Many things. I think people need to be aware that they can get infected with viruses, but also bacteria that are transmitted by animals. And, this starts with pets. This starts with dogs and cats, for example. There was an alpha coronavirus that jumped in Indonesia from dogs into humans and caused an issue there. It didn't spread, right? But this can happen. And so this is something to keep in mind. And then, of course, if you work on a farm, if you work in a slaughterhouse, it's always important to take precautions so that you don't get directly exposed or you have personal protective equipment. If you live in the south of Austria, you have to be aware that there is Pumala virus around, and there are there can be a large number of infections every year, that you have to be careful when you, for example, clean out your garage, because there might be feces from these rodents that transmit the virus, right? So I think just to make sure that the general population is aware of these things helps already a lot. And then, of course, if you have some kind of hotspot like avian influenza, and at the Altidona all the swans die, then you need to inform people that they shouldn't touch them, that they shouldn't go close to them, and so on and so forth. But in general, just raising awareness in the population that there is a lot of chance that viruses, bacteria might transmit from animals to humans, that would already go a long way.
Very important reminder. So, thank you very much again for the beautiful lecture and your precious insight. It has been a huge privilege for our university to have you today for the university lecture. I wish you all the luck and all the best to New York, and thank you very much for taking your time. Professor Florian Krammer. Alles Gute.
Herzlichen Dank für die Einladung.
Danke vielmals. Alles Gute. Damit schließen wir jetzt diese wunderbare Universitätsvorlesung ab. Ich hoffe, Sie hatten ein wenig Spaß und Interesse. Ich habe es faszinierend gefunden und sehr viel dabei gelernt. Ich glaube, Professor Krammer ist ein herausragender österreichischer Wissenschaftler und wir können auch ein bisschen als Österreicher stolz auf ihn sein. Wir werden jetzt eine kurze Pause machen und um 17:30 Uhr, das ist in ziemlich genau 30 Minuten, machen wir dann weiter mit dem Alumni-Club-Format Lebenswege. Es werden die Lebenswege von Ingrid Pabinger, emeritierte Professorin für Hämostasiologie, und der Lebensweg von Professor Anton Lagner, emeritierter Professor für Notfallmedizin, beleuchtet. Die Interviews werden geführt von den beiden Vizerektorinnen Fritz und Rita, und ich freue mich schon. Es ist auch sehr spannend, vor allem für die jüngeren Kolleginnen und Kollegen, zu sehen, wie so eine Lebensspanne in der Medizin funktionieren kann. Alles Gute bis dahin, nehmen Sie sich einen Kaffee und ich freue mich, Sie dann wieder um 17:30 Uhr bei uns begrüßen zu dürfen. Danke vielmals.